contact between the first neuron and the second neuron. So the electrical impulse can directly pass through the connection from one neuron to the another neuron. Whereas in earlier case, chemical synapse, there is a gap between one neuron and the other neuron. We will discuss more about a synapse in coming sessions. Right. So now let me discuss about types of neurons. So we can classify neurons based on the direction of communication. As I discussed in our earlier class, afferent neurons are those which collect the information, external stimuli with the help of a sensory organs like eyes, ears, nose. All these sensory organs, tongue, they collect the information and then they send the information to the brain. The neurons which helps in transfer of signal from the sensory organ to the brain are called as afferent neurons, also called as sensory neurons, because they are binding to the sensory organs. They are also called as sensory neurons. So the second category of neurons which helps in communication are called as efferent neurons. So efferent neurons are those the nerve cell that carry information away from the brain or spinal cord. That means the nerves signaling will be from the central nervous system to the effector organs like hands and limbs will get the signals from the brain through these efferent neurons. So whenever you want to do a task, if I want to raise my hand, my brain will send a signal from with the help of efferent neurons to my hand so that I can take an action. Or if my heart has to contract autonomously, so the autonomous nervous system will helps in contraction of cardiac muscle, or even the other muscle contraction also is directed by the efferent neurons. So interneurons particular only in local aspects of the circuit. So it's a transfer of signal from one region to the another region is done with the help of interneuron. So these are the three basic categories of neurons based on direction of communication. First one is called afferent neuron, the second one is called efferent neuron, and the third one is called interneurons. Right. So another important classification or divergence of neuronal cells are also called as types of neuronal cells. There are four major types of neuronal cells. First one is called astrocytes, second one is called oligodendrocytes, third one is called glial cell, and the fourth one is called microglial cell. Right. So astrocytes, so these are very branched structures which are generally present in the central nervous system inside the brain and spinal cord. They helps in rapid transmission of signal inside the brain. Oligodendrocytes are other types of neuronal cell, or the main function of these oligodendrocytes is to laminate the axons. That means we have discussed about lamination of axons with the help of a myelin sheet, right? That myelin sheet myelinating around the axon is done with the help of oligodendrocytes, right? So very good example of oligodendrocytes is squamous cell in case of peripheral nervous system. The squamous cells helps in myelinating all the neurons in the peripheral nervous system. And the third category of cells are called as glial stem cells. So these are, name itself suggests stem cells. These are stem cells present inside the brain. They helps in proliferation of the neurons whenever there is a neuron damage, or whenever there is a damage to the neurons, old neurons will be replaced by the new neurons with the help of a glial cell. So when the glial cell undergo division, glial cell will give one glial cell, daughter glial cell, and then a differentiated neuron, right? Differentiated neuron will be replacing the dead or damaged neuron. So similarly, next category of neuronal cells are called as microglial cell. So these are also stem cells, but they have limited potency when compared to the glial stem cells. So these are derived from hematopoietic stem cells which are present inside the bone marrow. So from the hematopoietic stem cells, the microglial cells are derived. They majorly helps as a scavenger cells to remove the cell debris at the site of the injury. So these are the major types of neurons in the nervous system. So have a look at this picture. Three major types. First one is the astrocytes, which I am talking about. More radiated cells, very branched structure, which are present in the central nervous system, especially in the brain. And so the second category is called oligodendrocytes, and these helps in the myelinating the axon so that the signal will be properly transferred in between the neurons, and there is no leakage of the electrical signal inside the axon and in between the neurons. And the third category, glial as well as microglial cell, both looks almost the same. Glial cells are the stem cells which are having maximum potency, whereas microglial cells will have a limited potency which are derived from the hematopoietic stem cell. So these are the structures, how they look like. Right. So let's have a look at the functions of the glial cells. Glial cells are essentially contributors to, for repairing the nervous system. As we are discussing, glial cells are the stem cells. They helps in repairing the nervous system whenever there is a damage to the nervous system. So they act as a stem cells in brain where they promote the regrowth of the damaged neurons. Also, another important function of the glial cell is they act as a scaffold for the development of the neurons. Let's say whenever in a particular region or in an organ a nerve system has to develop, the glial cell act as a scaffold or a template on top of which these neurons can grow, and then neurons can do the signaling. Right. And the last major important function of a glial cell is provide an interface between the brain and immune system. Right. So this is also an important function. So these are the major functions of glial cells. Right. So now let's discuss about another very important topic in CO1 in neurobiology, that is synapse. Right. So in the session 1 we discussed about what is a synapse. Synapse is a tiny space between two neurons. Right. So if you have a look at the picture, this is the presynaptic neuron and this is the postsynaptic neuron. Right. Both cases, this neuron as well as this neuron are presynaptic, this one and this one are postsynaptic neurons. That means this is the first neuron, let's consider this is the first neuron, this is the second neuron, first one, second one. So signal always transfer from the presynaptic neuron to the postsynaptic neuron. Right. So whenever action potential reaches to the terminal buttons, axon, we discussed about axon terminal buttons. So this is the axon terminal button. So whenever the signal action potential reaches here to this particular point, so the calcium signaling helps in vesicular transport of the neurotransmitter, and the neurotransmitters will be undergoing exocytosis and then they will be released into the synaptic cleft. So this space is called synaptic cleft, and the neurotransmitters like acetylcholine or GABA or dopamine will be released into the synaptic cleft, and then this neurotransmitters will move through the synapse and then it will bind to the receptors which are present inside the postsynaptic neuron. Right. So this is not a random binding at receptors, only a specific ligand can bind to a specific receptor. So this is GPCR, G protein coupled receptors, this one, and these are acetylcholine receptors. So acetylcholines can only bind here to this particular type of receptors. Right. And then once the signal is passed here, from here to here, the action potential continues here in the second neuron, and then it will move on to the third neuron, and then it will move on to the fourth neuron. Likewise, the signal will continue from one neuron to the other neuron. Right. So This is about chemical synapse, and what you see here is electrical synapse. So in case of electrical synapse, so there is no space in between the one neuron and another neuron, and there is a direct connection between one first neuron and the second neuron. So the electrical signal will directly move through these particular channels, the proteins which are integrin proteins which are present in between the two neurons, and then the electrical signal will pass through these molecules which are there in between these two neurons. So this is about chemical synapse and electrical synapse. So few important terminologies which I discussed just now. To summarize, presynaptic terminal, that means presynaptic neuron axon cleft is called a presynaptic terminal, which is going to release the neurotransmitter, and the synaptic cleft is nothing but the synaptic space which is there, and the postsynaptic membrane present in the postsynaptic neuron, that is in the second neuron, which has the receptors for capturing the ligands. Then other important terminologies like your neurotransmitters, important neurotransmitters like acetylcholine, dopamine, serotonin and glutamate. These are the important neurotransmitter released from the presynaptic neuron, going and binding to the receptors on the postsynaptic neuron. Synaptic vesicles nothing but a small tiny sacs which are released by the presynaptic neuron. These sacs will be carrying the neurotransmitter, and then they will be releasing this into the space synaptic cleft with the help of exocytosis. And receptor protein, so these receptors are generally present on the postsynaptic neuron, like we have seen GPCRs and N-acetylcholine receptors and other receptors specific for ligand binding, neurotransmitter binding, and neurotransmitter release also from the presynaptic neuron happens only when there is a signaling, only that is done with the help of voltage gated calcium channels. And next one, signal transmission. So whenever the neurotransmitter comes and bind to the postsynaptic neuron, the signal will be propagated from one neuron to the another neuron. So once the signal is transferred, whatever left out neurotransmitters present in the synaptic cleft will be reuptaken into the presynaptic neuron, and also they will be degraded, right? So these are the important terminologies in case of synapse, right? So you can see the pictures, and this is a chemical synapse where a presynaptic neuron and a postsynaptic neuron is there. You have the vesicles, you have the release of vesicles by exocytosis, and the neurotransmitter moving and binding on to the receptors present on the postsynaptic neuron, and then the signal is propagating from one neuron to the other neuron. So you see very tiny space between the two neurons, right? So this is about the chemical synapse, generally present inside the brain in the gap junctions. So there is a direct connection between one neuron and another neuron. If you consider this one as a presynaptic and this is a postsynaptic, there is a direct connection, and the electrical signal will be propagated here. IP3 and calcium can also move through these receptors. So this is about the session CO1. In 2 minutes I'll start the next session. Thank you.